I think it's good to see you all. Mary, you want to turn on this side? No, you're good. Okay, all right. And so welcome to our teams in Wisconsin. Primarily combination of several things, an NIH grant that basically brought about emphasis on glycosciences, and to a large extent it connected different campuses. So we not only have a program project, but we have a K12 grant application, which feeds into the R25 grant, which was the initial sort of source for generation of this particular course. And so I think it's very good, as I was telling you guys, but I think I'm not told the Wisconsin folks and the New York folks that we have several researchers from different parts of the country who will be lecturing in this course. And a quick question to the New York and Wisconsin people. Do you have the slides for this particular lecture?
No, no from New York.
Okay.
No from Wisconsin.
Okay, so I think what I'm going to do is I'm going to lecture for the first, let's say, 40-45 minutes. Then I'll walk to my office and send you all an email with that attachment. So hopefully you'll be ready for the next hour. Would that work?
Sure, thanks.
Okay. All right, very good. I'm sorry about that. All right, so essentially what is this course about? This course is about, so I can walk up, right?
Yep. I got you. Go ahead.
Somehow I like to walk around. So.
That's okay.
Okay, so essentially what is this course about? This course is about introduction to the fascinating science of glycobiology, right? This is about understanding glycans, and to a large extent, because of my particular training and focus on drug discovery, the idea here that we are trying to pursue is that we want to understand the role of glycans in biology, with an emphasis on trying to discover something interesting with regard to a specific group of molecules that might actually be useful in the clinic. So the idea is to think of glycans as drugs. And the reason why that is important is because, as you will see today, very much in this particular lecture itself, there are very, very few drugs that are made into the clinic as drugs. And although, as you would learn across the entire course, the glycans play a phenomenally important role in practically all aspects of processes, be it from growth and morphogenesis and differentiation and immune response and truckload of things. And it's practically left to our imagination to understand as to what could glycans actually do. You're going to see in the example that I'm going to present at least a snapshot of how relevant glycobiology is. Okay, so how do I advance?
I'll take care of it for you.
Okay, you take care of it. All right. Are you going to be here all lectures?
I'm going to sit right here.
Is it moving? No. Okay, so these are the learning objectives for the course, and as you can see, it's primarily about the overview of glycobiology, understand what glycans are, and this is primarily for this particular lecture, understand where glycans are located, the different types of interactions made by glycans, evaluate structures, and then think about glycans as drugs while learning about what molecules, proteins to glycans bind to, and have a snapshot of the type of molecular mechanisms that glycans work. This is quite an undertaking. Essentially, we are compressing a whole group of lectures into one sort of a very, very superficial view of the field of glycobiology and, in my opinion, glycochemistry. Okay, so this is the agenda that we have in front of us, right from what are glycans through the unique mechanism. Okay, can I get the next slide? All right, so let's think about what are glycans. All right, glycans, the word glycan has essentially come up into the fore fairly recently. Originally, if you really look at all the literature back at least 100 years, then you're going to find the word carbohydrate. Carbohydrate is the primary word that has been used And these days, there is a distinction between the meaning of the word carbohydrate and glycan. So carbohydrates are entities that for most part, are simpler units. Do you have it? I made a mistake in printing. Okay. All right. So carbohydrates are simpler entities. Generally speaking, they are monosaccharides, and for a large extent, the research on monosaccharides also took a fairly long time. There was actually a Nobel Prize awarded for research on monosaccharides. Simply elucidation of structure of glucose, for example, was worth a Nobel Prize. But the word glycan is typically, so monosaccharide, the word monosaccharide is also used by chemists. And monosaccharide also refers, carbohydrate also refers to people doing work essentially in an in vitro fashion outside of biology. Biologists typically like to use the word glycans. And for a large extent, because of how different structures of carbohydrates are there in our body and in general in the biological world, these are typically oligomers or polymers of the simplest monosaccharide unit. So glycans per se generally refer to oligo or polymers of specific type of sugars or composition of sugars. And yet, you're going to find that there is one particular monosaccharide which is now growing in importance, which is also defined as a glycan. That is N-acetylglucosamine, called glucan. And that is now becoming more and more important. And so people refer to that also as glycan, although it's a monosaccharide that is bound to a protein and it functions in a biologically clearly important manner. Now, before we go on, I have two structures that are clearly shown on the slide. And these two structures refer to what a sugar is or what a carbohydrate is. Okay? And to give you, because some of you are biologists, to give you a very simple way to really understand as to what would constitute a glycan. How do you actually know that a particular structure is glycan? Here is something that you should probably keep in mind. So first of all, generally speaking, glycans are cyclic structures. They have a cycle. On top of it, within the cyclic structure, you're going to find that there is an oxygen atom. Okay, so this is called a nuclear oxygen. Okay, it actually forms the cycle. And then, if you can identify all the different carbons, for example, carbon 1, and the numbering is fixed. So carbon 1, carbon 2, carbon 3, 4, 5, and 6. Then a sugar is that structure in which every carbon is connected with at least one oxygen atom. Okay, so that's the defining feature of a carbohydrate. You have a sugar, a carbon, connecting with at least one oxygen atom. So you look across all of these. So if you come across any structure and you want to know, and if it has a lot of different oxygens, and you want to know whether this is a sugar or not, or carbohydrate or not, this is a simple definition that you want to probably employ. You identify the carbons and then say, does it carry a sugar or not? Then there is within it, there is one particular carbon that is very special carbon. That particular carbon is carbon number 1. If you look closely, this carbon has actually 2 oxygen atoms and not 1, right? So this particular carbon has 2 oxygen atoms, and we define this particular carbon as an anomeric carbon, right? This particular carbon is an anomeric carbon. An anomeric carbon essentially sets the numbering in motion, 1, 2, 3, 4, 5, okay? So this is how you clearly identify as to what could be a sugar. Can I, any questions at this point? Yeah.
Are we going to use the words carbohydrates and glycans interchangeably?
So we are going to, for most part, we are going to use word glycan because this is more about glycobiology. When we discuss chemistry, we'll probably be using the word carbohydrate.
But they're used interchangeably.
Right. It depends on the context. It depends on the audience that you have in front of you. And then the next lecture, I'm going to be talking about polysaccharides, okay, which are essentially the common scientific terminology used for defining all of these. Okay, glycans dominate as far as the biology is concerned, as far as cells are concerned, as far as life is concerned. Glycans dominate, and they are typically present on cell surfaces. If you look at the electron micrograph of a cell, then you will find that the plasma membrane, for example, the microvilli, on top of it, you have a thick layer, a fuzzy layer. Does it say that? Yeah. A dense fuzzy layer, which is basically coating the animal cell body. And this is composed of glycoproteins. This is composed of proteoglycans. This is composed of glycolipids. And this particular thick layer is the one that is, for most part, anything that is outside gets in touch with the glycocalyx. So this is the one that can actually form a barrier. This is the one that retains a whole lot of water and keeps the system moist. And this is extremely important. This particular barrier is called glycocalyx. It's a substantial physical barrier, and it maintains tissue structure by capturing water as well as porosity, because it does allow things to pass by. In fact, as we understand today, everybody's glycocalyx is fairly unique. Okay? And to a large extent, the body uses the glycocalyx to distinguish self from non-self. What it wants to take in versus what it wants to throw off. And then there are microbes that try to overcome the barrier by mimicking the self so that it can actually enter. And I have a slide on that too. Any questions on this? Okay. All right. So if we zoom in even further, all right. So now we are going from a very, very broad view to something at a cell level. Then here is the model of what the plasma membrane would look like. This is the lipid bilayer. Embedded within the lipid bilayer are proteins. Obviously, some of these proteins, transmembrane proteins, some of these are ion channels, some of them are GPCRs, things of that sort. All of this you have heard, but you'll be surprised that nearly all of these proteins, especially in the mammalian cells, have on their surface sugar chains or glycans. And there could be multiple sugar chains, not just one. And they would extend out in space. To some extent, this would constitute the glycocalyx also. So there are glycoproteins. So these are called glycoproteins. Not shown here, for example, are the proteoglycans. And we're going to cover that topic in just a little while. Likewise, there could be glycolipids. So embedded in the lipid bilayer, you would have the lipid part, and then that would carry the glycan chain. Those are called glycolipids. And sometimes they are small, sometimes they could run into several residues, thousands of residues sometimes. So you would have quite a variety of glycans expressed or displayed on the cell membrane. These are the glycans that are recognized, for example, by microbes, bacteria, virus. These are also recognized by certain antibodies that we might generate or we might have. And then obviously we have certain groups of proteins that are called lectins that also recognize these glycans. So this is the glycobiology that we should be understanding, because these sorts of recognition mechanisms turn on genes that help build, that come up with a response, a physiologic response or a pathologic response. And this is the glycobiology that is now coming to fore. Yes, Sam?
So what is a vector actually?
Where does it come from?
Yeah.
So it's a protein that basically recognizes certain types of sugars. We have a section, we have a class of lectins. So you would get to know different types. They recognize sialic acid. They can recognize mannose. They can recognize galactose.
What is the like? Is there a protein on like another eukaryotic cell or something that would recognize it?
So, for example, you would have certain plants that would have lectins that can bind. You would have certain cells that might have lectins that would bind. Come from all different sources. Yeah.
May
Okay,
I
any
interrupt
questions?
just for a second?
Yeah.
Yeah, this is Himangi from Lao Lab. I was just wondering if you could repeat the question that was just asked. I couldn't very well hear it.
Oh, I'm sorry, I forgot. So I have a student, Sam. He basically asked us to something about lectins. What are they and where do they come from?
Okay, thanks.
Okay. All right, very good. You can also interrupt and ask questions.
Yes, of course. Thanks.
I had actually communicated that if you use a chat box, something will pop up, but I don't really see that chat box.
There is a chat box. Yeah.
Okay, you should be able to see. Okay, all right. So no chat says yet. All right, let's go to the next slide. Okay, so in terms of glycobiology, what is the focus? The focus is on this kind of recognition. Glycans have a number of roles, and we are not really discussing all the different roles. The primary role of glycans, at least up until several, I would say a couple of decades ago, was that glycans are structural molecules, right? For example, the reason, for example, we have these desks, right? I hope they're wooden, but if they're wooden desks, then what is that? That's basically all sugar. There is a particular type of sugar, polyglucose, which is beta polyglucose, which is all the sugar. The polysaccharide from glucose, which we can eat, is an alpha form. The one that forms structure. By structure, we mean strength. Okay? This is the one that offers strength. This is the one that provides things that can stand on its own. For example, another form of structure is, let's say, our bones and cartilage, right? So we have chondroitin, which is a major composition of the cartilage, and that provides structure. But in the last 20 years or so, more and more recognition roles are being discovered, and that's the primary focus of this series of lectures. The recognition arises, as I was telling you, from the identification of self and non-self. That's one major aspect. Okay? And so every cell, practically every cell, I would say all cells, typically express glycans on their surfaces, be in the form of proteoglycans or in the form of glycoproteins or in the form of glycolipids. These have certain structures. So you're going to see, to some extent, these different symbols with different colors being utilized. The base monosaccharides that form a glycan are just too many. And therefore, people have started resorting to different symbols, circles, diamond, square, with crosses with different colors to represent a code. And this is defined by the International Society that takes care of glycobiology.
I have a question.
And so, I have a question. So, if you move away from microphone, your voice fades away and noise builds up. Next tower is you can use a pointer electronically in presenting screen instead of the hand. Yeah, I can do that. Why don't you move away? Okay, can you get me a laser pointer?
Laser point.
So this will mean that some of you might not be able to see me a whole lot, but hope we can.
This is.
Let's try that also. Laser pointer. Yeah,
This one?
pointer. And then go on to that side.
So it gives.
Okay, so Christina is around. How does laser pointer work? Pen. No.
Let's try to.
Spotlight. Spotlight.
Yes. No, it's not working the way.
Okay, can you? So Chelsea, I don't know whether you can hear me, but I don't think the laser pointer is working.
Yeah, I could see your mouse when you're moving around, but yeah, either way is fine. I'll try to follow.
Okay, maybe I just need to stand behind the podium and not move. That'll require me to change a whole lot of my makeup. Okay. Oh. Okay, can you see the cursor?
Yes, it's way better.
Yes.
Okay. Wisconsin guys, can you see the cursor?
Yes.
Okay, perfect. All right, so let's try this then. All right, so every cell has, oh, this is just incredibly slow. Every cell has the glycans, okay? And these glycans can be recognized by extraneous microbes. M here standing for microbes, and they have certain proteins, that's what they recognize. Not only that, you might actually have, for example, a system where there is an internal negation of the signal. And this is how the self and non-self might actually work. So this is a very, very general diagram. Don't try to parse it out too much because this is very complicated. But the idea proposed by Ajit Varti of UCSD is that you could have all of these mechanisms at play in order for the biological recognition. And you realize for those individuals who are interested in discovering entities that could modify in clinical sense, that these glycans, or the structures of these glycans, appropriate modification of structures of these glycans would actually tinker with this kind of a process. And when that happens, you can modulate physiology or you can modulate pathology, whatever is the condition, right? So that's essentially the key part. Can I go forward? Okay, does this work? Oh, it works. All right, very good. Okay, so here is a very simple kind of teaser for you all to essentially highlight where all glycans have already been known to play a significant role. And by the way, this is a very, very short list because the list is pretty big, but I think this is simply easy to understand. We already talked about recognition in terms of microbes, so microbial infection, bacteria, viruses, and a bunch of other things. Immune responses, the way phagocytes recognize or macrophages recognize and kill, and then microbes. You'd be surprised that in the process of the first event for generation of life, where the sperm meets the egg, that is a glycan-dependent process. A very specific glycan is actually involved in the process. And the logic there to continue into the drug discovery domain is that if you wanted to prevent it, you could actually use that particular glycan to use it in a competitive fashion so that you won't get fertilization. Leukocyte recruitment, immune response, for example, the way chemokines, for example, move, glycans are actually involved to the site of infection, to the site of protection, things of that sort. Cancer, many elements of cancer, many hallmarks of cancer, for example, utilize glycans, a huge role for glycans. Metastases, for example, is one of them. There's a very nice paper that I'm going to showcase in just a little while that talks about how flowering actually utilizes a unique glycan species, simply flowering, and that's pretty interesting. And as, for example, all of you might already know that the reason why we have blood groups A, B, O, to a large extent, these blood groups are determined by the structure of glycans that are present on proteins that are lining our cardiovascular system. And that the difference is very, very small between blood groups A and B or blood groups A and O and several other blood groups. So these are very simple, kind of quick things that you might be able to point out if somebody was to ask you as to what are the common roles of glycans. So for these cases, there is absolute evidence that glycans are involved. Okay. All right. So now I'm zooming in even further and trying to go into the molecular structure of individual entities. For example, here is a glycoprotein, right? So typically most people think of proteins as being composed primarily of the amino acid chain, right? 100 on average amino acid chains, and they have unique structure, alpha, beta, things of that sort. They might be free protein, secreted protein, or they might be membrane-bound protein. Here, for example, is the GPI acyl chain, which inserts into the membrane. So this is a membrane-bound glycoprotein called Ty1, and this particular Ty1 has 3 glycan chains. As you can see, if you notice the movement of the cursor, although it's very slow, N glycans are written up. And so here is the entire volume of the protein, and then you can see the glycans as oriented in 3-dimensional space. They're actually going way out in 3-dimensional space, outside of the protein envelope. So you can clearly see that if something was to come and bind to a protein, there would be a glycan chain that it would encounter. If that doesn't play a role, it would have to push it aside and then seek the sequences of amino acid residue that it's trying to seek. But you might also have recognition arising from these sugar chains. You'd be surprised to know that although it simply says N glycan here, N glycan here, N glycan here, the structures of these 3 N glycans do not have to be the same. They could actually be different. And that's the key. There are probably a lot of different structures, not probably, there are a lot of different structures of N glycan, even if we say N glycan, on a particular protein. And that's where arises the heterogeneity. I don't know how many of you, yeah, go ahead.
So the substrate could recognize the N glycan instead of the protein sequence, instead of the amino acid sequences in the protein, and potentially cause changes.
That's correct. The substrate or whatever that entity is could recognize the glycan part alone too. But it will also recognize the amino acid, and generally, that's also equally true. I don't know how many of you are actually following a whole lot of debate, that is not debate, regulatory discussions that are currently going on with regard to the biosimilars. Say, for example, a particular protein, which is the glycoprotein that enters the clinic, and it's a glycoprotein, then in terms of what constitutes the glycan part becomes very important for a generic drug manufacturer to put its product into the clinic. Why? Because the generic or a biosimilar, generic is not the word used for biologicals, but the biosimilar product has to essentially match the composition of this glycan in addition to absolutely matching the identity of the protein. So that becomes extremely important. And as I'm going to show you just a little while, there are thousands of glycan sequences that are possible within the domain that we are talking about. There are multiple sites. Remember, there are only 3 N glycans shown here, but the number of asparagines, N glycans come from site of labeling, which is asparagine. The number of asparagines that might actually be present on the protein might be more than 3. Okay, so those are not labeled. And in terms of the biosimilar, it's extremely important to figure out where is the glycan chain. So therefore, pharmaceutical development is extremely important. Recognition, obviously, is also extremely important. So biology is also extremely important. Any questions at this point? Okay.
You mentioned asparagine. I didn't understand what asparagine is and does.
Asparagine is an amino acid.
Yeah.
And basically, asparagine's side chain, CONH2, that N is labeled with the sugar, the glycan. Okay? So this is a chemical modification that arises, and it could be one sugar, as I was telling you, with N-acetylglucosamine. It could be multiple sugars. It could be linear. It could be branched. Okay, can I go forward? All right. Glycans present phenomenal structural diversity. I'm going to basically separate glycans into two parts, although there are actually way more than two parts, but simply for two major sections. For simplicity's sake, I'm going to separate those into two sections. One is the neutral sugars. In the neutral sugars, although they are not technically only neutral, we are including sugars such as glucose, galactose, mannose, xylose, fucose, sialic acid, all of those. Sialic acid has a negative charge, but for simplicity's sake, I'm including that in the neutral sugars. There are many different types of monosaccharides. In fact, there are too many to even list, but let's say these are the most common. Within the category of hexoses, which is basically 6-carbon unit, you're going to learn about these in just a little while. There are 16 different types of hexoaldoses. There are 8 different types of 6-membered ketoses. There are 8 different types of pentoses that are aldoses and things of that sort. So if, for example, you were to consider just glucose as a base scaffold and try all possible modifications around that, which includes galactose, mannose, iodose, all of those, then you're going to come up with 16 different types of these. And so you can imagine that if you have 3 sugars multiplied by 16 at each position, the number of sequences that you can get is absolutely phenomenal. That's what nature exploits in trying to evade microbial invasion. Okay, and this is fairly common. There are 2 common sialic acids, which is called neuraminic acid and KDN, and plus there are numerous others in real life. And if you simply compare all of these base units with what we have from amino acid, there are 20 common amino acids. There are way more than 20, but there are 20 common amino acids. And there are essentially 8 nucleic acids, both DNA and RNA type. So as compared to that, the numbers here is absolutely phenomenal, which is the reason the glyco world is incredibly diverse. In fact, it's even if you just consider the neutral sugars to a large extent. And so this is from the base common structure, but from the way in which the glycans are connected with each other. So now if you start thinking about that, when one glycan is connected to another glycan, let's say from position 1 to position 4, is different from essentially same scaffolds or same monosaccharides connected from each other from 1 to 6. So that's completely different, and it's a unique structure that is generated. And likewise, there are so many other possibilities that arise from 1 to 2, 1 connected to 3, and these are all interglycosidic between 2 sugar linkages. So the number of possibilities that keep increasing, and if you now consider compare it with, for example, proteins. Protein have essentially 1 type of inter-amino acid bond, which is the amide bond, or the peptide bond. And nucleic acid essentially have 1 type of inter-nucleoside bond, which is the phosphodiester bond. So there are just way more possibilities with regard to sugar. Way more possibilities imply, hopefully, nature just doesn't produce things for useless purposes, right? So if it exists in nature, most probably it serves a function. It is for us to essentially understand as to what that function is. Now, let's consider substituents on top of, what are the base scaffold. For example, for proteins, we know that you might actually have different post-translational modifications. Well, glycosylation is also a post-translational modification for proteins. But within sugars, you can actually have several different types of substitutes. It could be an amine, it could be an amide, an acetyl group, it could be an ester, which is a sulfate, but it could be a phosphate. It could be deoxy or oxy, and so many others that are possible. And this adds to the structural diversity of the glycans that are present in nature. Now, aside from the neutral sugars, there could be acidic sugars. Okay? And these acidic sugars are quite a lot. And these are, for large part, all known by the term glycosaminoglycan. So these are all GAGs. glycosaminoglycan, I'm showing you one example of glycosaminoglycan, which is heparin and heparan sulfate, which have similar base scaffold. But it's a polymer, as you can see, and this polymer is uniquely connected between carbon atom 1 and carbon atom 4 through an oxygen bond. And the classic feature of this, not to go into too much detail, is that this particular molecule has a number of negative charges. So it is highly acidic. In fact, the pKa of this particular sulfate group is 0.2. And therefore, even in stomach, which is a pH of 2, this particular entity, this particular moiety would be negatively charged. Okay, so even in stomach, this would be negatively charged. So this is the most acidic biomacromolecule that is known, and it exists in our body. And as far as heparan sulfate is concerned, it is there on practically all cells in the form of proteoglycan, and we'll see what that is. To give you an idea about the structural diversity of glycosaminoglycans, if you consider the heparin or heparan sulfate glycosaminoglycan, then there are 48 different types of disaccharide. I can highlight it too. Very good. There are 48 different types of disaccharides. And if you consider that we have a hexameric unit, right? We have a hexameric unit, then 48 different types of 48 raised to 6 are the distinct possibilities of the number of structures of a hexameric form of heparan sulfate is there. Heparan sulfate that we have could actually have more than 100 monosaccharides in a chain, but we are simply considering 6, which is 12 residues, hexameric unit. And the number of possibilities are 50 raised to 10 raised to 12 unique structures are feasible. If you compare with nucleic acid has 4, right? A, G, T, C. And therefore, you would have 4,000 unique sequences. If you consider proteins, there are 20 residues. That brings up the total to 64 million. But here it reaches into billions. And you can imagine as to what our body, and for that matter, what our cells might actually have. For that matter, nobody as yet truly knows the gagome or the glycome of an animal body, the entire number of different structures that might be feasible, and it's a huge project. Some of these sequences are already known to modulate function, okay? So you're going to see that antithrombin binding heparin pentasaccharide is actually used in the clinic as an anticoagulant. And likewise, some of the research from my lab as well as other people's lab has shown that there are some very unique sequences, not many, but very unique sequences that modulate function. And so those sequences might actually become very useful for clinical purpose, both as probe to understand how biology works, as well as hopefully to help relieve certain symptoms or diseases. Okay? Any questions? All right. Now, let's quickly, what time is it? 3:45? Okay, so let's go for 5 minutes and then take any questions if you have, then we take a break. In the meantime, in that break, I'll send the PowerPoints to both of those sites. Okay, so what I want to do is I want to quickly highlight some of the drugs and glycan-based drugs that have actually reached the clinic. One of the major ones are anticoagulants. Heparin-based anticoagulants for students of pharmacy. This is obviously quite a repeat, but others might not know about it. The drug that is currently used in the clinic as an anticoagulant, a small molecule glycan, which is Fundamentals. It is a 5-residue sequence, as you can see from here, and it basically binds the protein, as shown here, at a specific site, a protein called antithrombin. Antithrombin is a natural anticoagulant that we all have, without which we cannot survive. And this particular pentasaccharide activates antithrombin to become a much better inhibitor of coagulation proteases, which is why it is used as an anti-clotting agent. Nowadays, this Fondamentals is completely made synthetically. It is fully homogeneous. It's fairly expensive. Another class of drugs which are very heavily used, especially these days with the sudden temperature changes and the infection that is currently going on, lots of people are suffering from the flu, is Tamiflu. Tamiflu is actually oseltamivir. And this particular drug is not really a sugar. It is a mimetic of sugar. Okay? The real sugar is zanamivir, which is also a drug which had been approved, but up until the time Tamiflu came along, zanamivir was the one that was heavily utilized. This molecule, which is a sugar, actually mimics the sialic acid structure, actually mimics sialic acid that we all have. The reason why it mimics sialic acid is because sialic acid is the one that is recognized by the enzyme neuraminidase. So by the enzyme neuraminidase. And so neuraminidase binds to glycans that contain sialic acid, and it cleaves it. And one of the mechanisms for release of influenza buds, influenza virus, is this particular cleavage mechanism. That once it enters the cell, it undergoes a whole lot of amplification, a lot of viruses are generated. They come back to the surface of the host cell, and from that point, at that point, they're attached, but then they are released into bloodstream by the action of neuraminidase. And so if you inhibit that, the propagation of the virus will be prevented. And this actually dictates therapy. The way in which Tamiflu is used is highly dependent on this particular mechanism. At what point would you want to take Tamiflu? There is a recommendation for that. Oops. Okay, so that's another very big market. I think the market cap is somewhere in the region of $2 billion or so of Tamiflu, quite a lot. Now, the reason why that is because there are certain proteins that are recognized by these different glycans, and once again, we can divide it into two different, significantly different types of carbohydrate-binding proteins or glycan-binding proteins. Those are the lectins. There are several different types of lectins. They originate from several different sources, plants being the major source of these lectins. There are plants that are lectins that are called L-type lectins, C-type lectin, R-type lectin, I-type lectins, P-type lectins. I-type lectins are also called Siglecs, sialic acid recognizing lectins. P-type lectins, you're going to see somewhat about mannose-6-phosphate and the role of mannose-6-phosphate in certain receptor in certain diseases. And there are galactins, which is galactose recognizing lectins. And unfortunately, there is no universal classification of lectins, although people are hard at work. So this is historical. Researchers have identified these different lectins to do different things. They recognize different things. And from that is the name derived. And so that these different names have propagated. And so I think those continue to be used even today. The second major class of glycan-binding proteins are the GAG-binding proteins. So these are recognizing heparin, heparan sulfate, chondroitin sulfate, gametan sulfate, things of that sort. And there are a huge number of proteins. Without going into too much detail, I'm going to be talking about, you're going to listen to, for example, growth factors and the receptors in a lecture. You're going to listen to antiviral lecture, which is where viral envelope proteins will come into play. You will listen to chemokines and neutrophils. There's a lecture on that you'll be exposed to. I'm going to be talking about coagulation. There are enzymes in coagulation that you're going to talk about. Serpins and enzymes will go together. But just to give you a brief idea, there was a paper published in 2011 which essentially simply mapped the glycan or GAG binding property of all the transcripts that are expressed. They didn't really focus on the protein and the binding part, but they focused on the transcripts. And they identified 400 different, they proposed 400 different proteins are recognized by GAGs in our body. So that's quite a huge number. Obviously, nature would simply not come up with something of this sort. There has to be a reason. and it's just for us to figure out as to are there any specific sequences that are involved in this recognition, and therefore can we convert that into a probe or a drug. Okay, let's see, this is slide number 6. This is a very short slide. So here are the glycan-based drugs that are currently used in the clinic. I've included primarily all are glycans, of which the anticoagulants, not just fondaparinux, heparin, low molecular weight heparins are all part of it, zanamivir. There are antibiotics, which most people don't think of glycans, but they actually have quite a pretty good glycan scaffold, neomycin, kanamycin, streptomycin, things of that sort. Congestive heart failure drug, the steroid is the active ingredient, but without the glycan or the glycoside, it just doesn't work. Digoxin, digitoxin, things of that sort. There is sucralfate, which is sucrose octasulfate. It's an anti-acid which has been approved. Not many people use it. There is a good diuretic, mannitol. Mannitol is a carbohydrate. It's not a cyclic structure. It's a linear structure. There is an anti-arrhythmic, anti-arthritic agent, which is glucosamine, chondroitin. You can see this in commonly used by elderly people. Acarbose is an anti-diabetic. It prevents the absorption of glucose because it inhibits alpha-glucosidase. Miglitol is also similar, inhibits alpha-glucosidase. There are anti-cancer agents, especially so many drugs that are currently in development. Hopefully those will reach the clinic. And there are several other indications for which hyaluronic acid is being developed, especially osteoarthritis. Okay, so you can see that if the interactions are so, if the library is such a huge library, if they play such an important role, the number of drugs that have currently reached the clinic and benefit mankind are very, very few. And that's the primary part. What the NIH wants to do is, NIH wants to stimulate research in this direction so that hopefully we discover something interesting which can actually reach the clinic. Okay, I'm going to skip this part because I'm going to do this later on when I do coagulation. Okay, so let's finish lecture 1. I'll take questions if you have any. All right. So yeah, I went really fast over the material, but hopefully, if you have any questions, even at a later point, you should either see me or send me an email so that I can cover, address the questions. Yeah.
Do you have like actual scheduled office hours?
You can, if I'm in my office, you can walk in most of the time. So, no real scheduled hours, but it's difficult for me to just set aside 1 hour. Okay, and then nobody shows up. Right? So, but you can always make an appointment. Just send an email saying, "I would like to meet." All right. So we're going to go to the fundamentals. All right. So this is going to be a relatively relative slog in terms of because this deals a whole lot of structure, and I can tell you, most people don't enjoy structure. So let's begin, right? But hopefully, I'll keep you interested. Okay. So what are the objectives here? The objectives are to understand as to how glycans are named. What are the different constituents of a glycan? There are several different representations. So I think you need to know different representations. At least in the literature, there are 2 different types that are used, and then the symbols and the color symbols that I told you a little bit about. Those also you need to know. What are the conformations these sugars adopt in solution? And then why do glycans conformation matter? Because obviously every little change in structure is going to determine recognition. And just to reiterate, this particular course is a whole lot about recognition. So, okay. So these are the different specific topics that we'll be considering. And let's begin with this particular slide, which at least a couple of you have probably heard me talk about, because this is absolutely fundamental, and I present this particular lecture to several different programs, not just in pharmacy, but also in chemistry, also in nurse anesthesia. So several different programs are presented. Anyway, starting from the word carbohydrate. The word carbohydrate essentially implies hydrate of carbon. And that's how back in the olden days it used to be, because those days structures were not very easily determined. But the earlier sugars that were identified followed this particular logic, that practically every sugar was a hydrate of carbon. What do I mean by that? If you have six carbons, then if you have this particular molecule, then this molecule, which is a glucose, can be represented as the hydrated form of 6 carbons, right? And likewise, sucrose could be represented as the hydrated form of 12 carbons, and likewise. So, and then the word stuck. It basically, people started using it. Later on, obviously, this did not hold true, because there were sugars that had a nitrogen, and there were sugars that had a phosphate, phosphorus, and things of that sort. So, but still, we continue to use it. And so the way in which you're going to identify whether a particular structure is a carbohydrate is the definition that I gave you in the first lecture, which is every carbon has to have an oxygen, or now we're going to revise it, every carbon has to have an electronegative atom. So nitrogen is an electronegative atom. So CN bond, for example, glucosamine CN bond would also satisfy the same definition. Carbohydrates are also, especially for carbohydrates, they're also typically used with certain suffixes and prefixes. So glucose, O's is a suffix. Sucrose, O's is a suffix. And likewise, just as we typically use for enzymes, we typically use the suffix ase, right? Glucosidase, for example, is an enzyme. Likewise, for carbohydrates, we use O's. And then there are prefixes. So we can use triose or tetrose or pentose, things of that sort. You have to remember that triose doesn't really mean trimer of a monosaccharide or a mono sugar. Triose means 3-carbon sugar. A sugar chain having 3 carbons. Tetrose means a sugar chain having 4 carbons. Pentose is 5 carbons and things of that sort. Okay. Now, this is coming from obviously from the chemical world, that there are 2 different types of moieties or functional groups that are present on carbohydrates. Carbohydrates can either have an aldehyde group, or it can have a keto group. So those that have an aldehyde group are called aldoses, and those that have a keto group are called ketoses. And if you're bewildered at this point, do not worry, I'm going to clarify that in just a little while. Okay, let's start with the simplest of carbohydrates. The simplest of carbohydrate has to have a 3-carbon atom configuration. You can't really have a carbohydrate from 2 carbons, although aldoses are clearly possible. They generally have this formula, CnH2nON. Common monosaccharides have 3 to 9 carbons. Saccharide essentially means a sugar residue. And so this is an aldose. It has an aldehyde group, right? This is the aldehyde group. The aldehyde group is typically always, not typically, is always present on the terminus. The aldehyde group can never be part of a chain, in the sense, can never be in the middle of the chain. So therefore, that's one major group. Then there are ketoses that are, how can I highlight this? Okay, oops. So then there are ketoses, and this is the keto group. So keto group can never be at the terminus, can never be at one end. Keto group always has to be in the middle. And as far as all natural carbohydrates are concerned, keto group is always present at the 2 position. is always present at the 2 position. Okay, that's by definition. This is how we have defined it. We gave this a numbering and that's it. And the simplest one is dihydroxyacetone. Okay, that's a carbohydrate. Why is it a carbohydrate? Because if you really look at this carbon is connected to an oxygen. This carbon is connected to the ketoxygen. This carbon is also connected to this particular oxygen, and therefore it is a carbohydrate. Okay, everybody with me? Yeah. Okay. Okay. Very quickly, I'm going to show you as to how diversity arises. I gave you a big, 10-minute session on phenomenal structural diversity of carbohydrates, and this is the foundation, right? And realistically, you simply have to follow the logic. You don't really have to remember anything in particular. But glyceraldehyde, which is an aldose, right, a trialdose, has a central carbon, which is a chiral carbon. What it means is that there are 4 different types of substituents. So therefore, it is called chiral carbon. And whenever there is something of the chiral carbon, it typically has 2 stereoisomers. Okay, these are completely different. They are distinct in several different properties, especially recognition. Recognition is highly dependent on the stereocenter. So in 3-dimensional space, they are different. So glyceraldehyde is therefore 2 forms. Okay, they could be an R form or an S form. So for the time being, I'm not going into details about what these are, but just take it from me. So through some rules, which are called Kahn-Ingold pre-log rules, we have defined these 2 different forms. One of them is an R and S. And so these 2 are different structures. These 2 are different structures in 3 dimensions. So this is the R glyceraldehyde, this is the S glyceraldehyde. And they are recognized by different, especially by proteins, which are also chiral, completely different. And then this particular structure, because it's difficult to draw, there are several scientists who came up with different types of representation. So here is a representation in which you simply draw this kind of a square, position all the 4 different groups, they're completely different, which actually is equally, essentially same as this particular representation. And likewise, you can have from the S side, you can have something of this sort. Now, this kind of representation had its own scientist, and so there are D and L forms, and there are R and S forms. So technically, these are completely different. The R and the S forms are simply derived through assignment of certain priorities. The D and L form are derived based on how you draw. And that's important for you to realize, because D-glucose is the one that is very, very sweet, is the one that we can metabolize, is the one that is present in starch, for example, things of that sort. L-glucose, on the other hand, is essentially completely impervious to our metabolism. It simply bypasses everything. It is not recognized by anything. And if you were to consume L-glucose, it would straight end up in feces. Okay, there's basically nothing. It would not be absorbed. So there is a striking difference between D and L chains, between D and L sugar. Just as for proteins, obviously there is an R and S form, and proteins of which you all have one specific form. Likewise, for carbohydrates, we generally have D form of a particular sugar, glucose, for example. On the other hand, for another monosaccharide, we have an L form which is present. So it is not uniformly D in our body, nor it is uniformly L in our body. Some sugars are D and some sugars are L. So, but how is D identified? So the way that it works is that when you write the OH group on the right-hand side, as here, so the first rule is that you have to have the aldehyde group at the top. This is simply writing. This is simply representation. This is simply to understand what structure is it and differentiate. So you can draw with an aldehyde group at top, and then you can write the different substituents either on the right or on the left. So when the OH group is on the right, the scientist who gave this designation is Emil Fischer, and he basically said, this is D. He just arbitrarily gave it a number. D standing for dextro, standing for right. That's what this is. The OH group is on the right. And likewise, the OH group is on the left. So he basically said L standing for LEVO on the left. Okay. So if you understand this, then hopefully things will become much clearer. So Emil Fischer basically assigned it. Now it turns out that there is a third way in which we can find out as to what the structure is. And back in the olden days, it used to be optical activity, in which you can rotate the plane of polarization either to the right or to the left, or plus standing for right and plus minus standing for left. Okay, so this was experimental measure, and he did not know at that time, but it so turned out that what he wrote as D actually turned the plane of polarization towards the right, and therefore dextro. So actually it matched. This was completely fortuitous. But it doesn't have to be the case for some other sugars, but it did turn out to be, at least in this particular case. Okay. So now, if that you've understood, then I think it sets into motion how the different sugars can arise. So I'm going to quickly point out the entire series arising from D-glyceraldehyde. So why is it D once again? Because the OH is on the right. And now if you go to a 4-carbon carbohydrate, this is the 4-carbon carbohydrate, then the nomenclature is that this particular carbon, which is the differentiating carbon, which is the one that basically says I'm a D chain or I'm an L chain, that has to stay exactly the same. Therefore, we keep it constant. And we simply keep adding other carbons on top. So if you go to 4-carbon, then you have to add a carbon between this aldehyde and the carbon here. That's this carbon. This carbon can have an OH on the right, or it can have an OH on the left. Okay, so you end up with two. Both of these are diastereomers. This is D-erythrose and this is D-threose. And if you continue in this fashion, if you want to go to the 5-carbon carbohydrate, sugar, then you end up with addition of this particular carbon in between this carbon and that carbon. So now you end up with an OH once again on the right, OH on the left. So these two, ribose and arabinose, arise from erythrose, and xylose and lixose arise from threose. Okay, and likewise, if you simply continue to 6-carbon monosaccharide, which is usually the most common one, ribose can give you allose and maltose. Note all of these are D. Arabinose can give you glucose and mannose. Xylose will give you glucose and idose. And lixose will give you galactose and talose. And all of these are completely different. All of these are unique, structurally completely different. And actually, as I was telling you earlier, there are lectins, proteins that recognize only glucose, or only mannose, or only galactose. So in biology, there is recognition, although you might think that these are all very similar, but actually in biology, there is phenomenal recognition. So these are the 8 monohexoses, right, arising from D. So how many monohexoses will you get from L? Exactly 8. So that constitutes the 16 that I was mentioning to you earlier, that these are the 16 D and L different forms that might be present. And you can see that even at the monosaccharide level, you have so many base scaffolds. So you can imagine that if you go to modified forms and then on top of it variations around that, you will end up with a phenomenal number of sequences that might actually exist. Here is a very simple illustration of what would happen for ketosis. For ketosis, we basically have the keto group at position 2, which is achiral. So we can't really have two structures. So there is no D and L form or an R and S form. So the first one that comes up is erythrulose or L-erythrulose here, simply for the sake that I want to show for L, in which the OH is between the keto group and the last carbon. And this cannot really change. Why? Because we have now identified that to be as L, and it has to remain on the left. So erythrulose will give you xylulose and ribulose by addition of that particular carbon. And then xylulose will give you psychose and fructose by addition of that particular carbon. And ribulose will give you starbose and tagatose. Okay, going on D and L. So you end up with 4. So just as you have 4 L hexoketoses, now we have ketose, we don't have aldoses. Likewise, we would have for D, you would have 4. So that generates 8 unique ketoses, at least for the 6 carbon scaffold. Okay, so these are the different monosaccharides that are feasible, and if you multiply with the chains and all the different possibilities, you'll get a large number. Very quick information for you all. D-glucose is called dextrose because it actually rotates the plane of polarization. By the way, before I go to this kind of information sheet, with regard to structure, any questions? Okay, any questions from Wisconsin, New York, DC?
No, but it's great. I like it.
Okay, thank you. All right. So, D-glucose is dextrose. When somebody is ill, really requires a lot of energy, quick IV, they actually give a dextrose drip. Actually, I have it here. IV drip that contains 5% glucose and then 15% saline. D-glucose is the most abundant monosaccharide. Okay, most abundant monosaccharide, and it generally D-glucose is present a number of times in fruit juices all by itself. Okay, all by itself. So D-glucose is an energy booster. Right away, you get significant energy. We have to have D-glucose in our blood quite a lot, 65 to 100 milligrams per deciliter of blood. And the tests are essentially designed to detect that. Fructose is another monosaccharide. Fructose is a monosaccharide that is present in disaccharide glucose. And then galactose is part of lactose. Monosaccharides, although I have shown you as typically linear form, remember the first slide? Yeah, go ahead.
Is dextrose so is it just linear D-glucose when they give it to you, or is it cyclic?
It is cyclic.
Okay.
Yeah. So normally the linear form would not really exist. The equilibrium is far on the right. Okay. The equilibrium is far on the right. When I say far on the right, this is the equilibrium that I'm talking about. Okay, so the linear form is the one that I've shown you so far, but actually linear form is not present to a large extent at all. In fact, 98–99% of the equilibrium lies in the cyclic form. Okay, why is that? Is because of this particular phenomena. Okay, so I'm going to just highlight what happens for sugar and skip the foundation in organic chemistry. So what happens is this is glucose, right? Look carefully, this is glucose. And in this glucose chain, which is the open chain form, we have the aldehyde group, and then we have the alcohol, which is somewhere around here. Well, there are quite a few alcohols, but as the chain folds in real life, this particular OH, the one that is highlighted, is the one that comes close to this particular OH, that is the one that comes close to the aldehyde group. I can't really pinpoint it. Oops. Is the one that comes close to the aldehyde group in three-dimensional space. Okay? And what happens is, because of this, the OH group can either attack the keto group from the top or from the bottom. When it attacks from the top, you get an OH group on the bottom in the cyclic form. When it attacks from the bottom, you get an OH group on the top in the cyclic form. So now you end up with an additional chiral center, which is 1. This particular carbon is now connected to 2 oxygen atoms. And as I was telling you earlier, this is the anomeric carbon. Sorry, this is the anomeric carbon. This aromatic carbon can either have the OH at the bottom or at the top. And therefore, we end up with two additional sort of nomenclature symbols, alpha and beta. Okay, so we have an D-glucose, but then we also have an alpha D-glucose and a beta D-glucose. The way it's worked, and this is simply by definition, is that if the sixth OH group and the OH group at anomeric position, this one, they are on the same side, then that is called beta. If they are on the opposite side of the chain, they are called alpha. So this is the definition. It's a derived definition. The actual definition is slightly different. It originates from the orientation of the C5 position, but I'm giving you the operational definition. If you want to quickly identify whether it is an alpha sugar or a beta sugar, you simply have to look at the position of the 6 carbon and the anomeric OH. If they are in the same plane, that's beta. If they are in the opposite plane, that is alpha. So this is how you define the anomeric carbon. And generally, as you can see, this particular equilibrium lies far to the right. Okay, this particular equilibrium lies far to the right. So maybe I should just, energize you a little bit to connect it with therapy, because this might be a little boring exercise. So I want to ask you a question. So, you all know about the glucose test, right? The glucose test measures glucose, okay, in blood, right? And one is obviously you measure glucose, could be 80 to 120, actually even narrower range, and you would say you are fine. But there is another glucose test, which is typically used to identify who is either pro-diabetic or diabetic, and that test is called the commonly used. You're all very young, you haven't undertaken that test, I know that. A1C test, okay, or HbA1c test. What is HB? HB stands for hemoglobin. And we are basically detecting hemoglobin's modification at position 1. Okay, at position 1, alanine position 1. C standing for carbohydrate. Okay. I hope that's the definition. But basically, glucose is modifying the N-terminus of hemoglobin molecule. Okay, glucose is modifying the N-terminus of hemoglobin molecule. Now, where is that coming from? That is basically coming from a reaction of the aldehyde group. Somehow it is not working on the aldehyde group. Okay. The aldehyde group at position 1 and the amine. And there is a specific reaction in organic chemistry, which some of you might have undertaken, called Schiff reaction, which is the reason why we have the glycated form of hemoglobin actually happening in our blood or plasma. Okay? Now, think of this particular equilibrium, keep that in mind, and then answer the question that I'm going to ask. So if you really look at the A1C level as the primary differentiator between people who are pro-diabetic and who are diabetic, then most physicians use a cutoff of somewhere in the region of 6.5 milligrams per deciliter, as a cutoff, 6.57, depending on physician that you're talking. How many milligrams of sugar, how many milligrams of glucose should be there in our bloodstream on a normal level? Well, you already saw that slide. 100, let's call it 100. 100 what? What are the units? Milligrams per deciliter, right? Now, 100 milligrams per deciliter is normal, but the A1C test or HbA1c test says that if you have 6.5 milligrams glycated form of hemoglobin present in a bloodstream, the person is almost pro-diabetic. Much higher than that is diabetic. So if you already have 100 milligrams present, how is it that only 6.5 milligrams of hemoglobin is going to give you that reaction? In effect, what am I asking? Why is it that the hemoglobin has to be modified only 6.5 milligrams? Why such a small amount of hemoglobin needs to be modified? And you would say, is pro-diabetic or diabetic?
Because the reaction depends on the straight chain form of glucose, and there's very small concentration in straight chain. So
Exactly. So when the reaction is dependent on the aldehyde group, this particular form, the cyclic form, cannot really react. So in our bloodstream, you had to have sufficiently high level of sugar for a prolonged period of time to basically have sufficient aldehyde group react with the amine. And that will give you 6.5 milligrams. So essentially, HbA1c test depends on this equilibrium, that's number one. Not only that, it also depends on this particular high level of sugar to be maintained over sufficiently long period of time to give you that level. So it's actually giving you a readout of several weeks, actually months. And not just a spike. You might have a spike, but it will not give you very high HbA1c level. Got it? Okay. So you can see that there is very fundamental basis for the tests that have been designed, chemical basis. Okay, so just as we have for aldoses, we also have for ketoses. So for example, for fructose, which is a ketose, it's essentially the same type of equilibrium that happens, but because the keto group is present at the 2 position, we get a 5-membered ring here. We don't get a 6-membered ring, as in glucose or mannose or galactose. We get a 5-membered ring. Right? Got it? Okay? But it's essentially the same thing. You get an alpha D fructose and a beta D fructose. Beta, in which the 6th position and the OH are on the same side, and then the 6th position and OH are on the opposite side is alpha. Okay? Same rules essentially apply. Okay, why should we worry about these different forms? Alpha, beta, things of that sort. We should worry about these different forms because these different forms assume different conformations. Okay, you have an alpha D glucose and a beta D glucose. In real life, the 6-membered rings exist as chair, and although to you these might seem to be essentially not important, they turn out to be important. Why? Because the polymer of beta D glucose is completely non-digestible. We cannot digest it. But the polymer of alpha D glucose, we can digest it very easily. So this is essentially potato starch, which we consume on a regular basis, which we really like. Right? Beta D glucose is paper or wood, things of that sort. So this is structure, actually, right? And so chair conformations and the orientations are extremely important. The other point is that this also dictates which is the dominant form under biological conditions. All right. So out here, for example, beta D glucose, you see, for chemistry-oriented students, all the substituents, the OH groups and the CH2OH are all equatorial in position, and therefore this is the most stable form. Whereas for the alpha, the OH group here is axial. So it is slightly less stable than the beta D glucose. And you can apply the same analogy to other sugars, mannose, galactose, and sialic acid, things of that sort. Okay? All right. So likewise, you can draw the cyclic forms of the galactose, mannose, and these cyclic forms of these kinds of sugars are all, for most part, shared in configuration, except for a few monosaccharides. For example, iduronic acid, which is not a chair. These monosaccharides typically undergo significant level of interconversion, which is called as mutarotation. Okay? So the alpha and the beta form can actually interconvert through the open chain form. So the anomeric carbon opens up, it gives you the linear form, and it gives you the beta, and this cycle keeps going back and forth. And the reason is, and this is the reason why you have small amount of the aldehyde in our bloodstream all the time, and that's the one that reacts. Okay, just to give you a quick orientation towards what you might actually see in the literature in terms of glycans as reducing sugars and non-reducing sugars, or reducing end and non-reducing end. Where is that coming from? That's basically coming from the ability of these different sugars to react with different types of reagents. For example, Tollens test detects aldehyde. The aldehyde gets converted into carboxylic acid. So it gets oxidized. In the process, the silver plus that is used in the test is reduced to metallic silver. So it reduces Ag plus to Ag0. And that is the reason why aldoses are considered as reducing sugars. So therefore, position 1, which is the anomeric position, is the reducing end. The opposite side, which is carbon 4, is the non-reducing end. Got it? So there is reducing end and there is a non-reducing end. And I'm going to skip this uronic acid up until the time we come to heparin, because we'll be doing it there. Okay, what time is it?
4:47.
4:47. 37. Oh, okay, so we do have time. All right, so I'm going to skip the uronic acid till the time we do heparin, where we'll do a little bit of uronic acid and conformation. So here are the different modifications in terms of orienting you in the biological direction. If you hear the term glucosamine, manosamine, galactosamine, and so many others, the amine is always at 2 position. This is given by biochemistry. This is given by nature. Okay. Unless it is a synthetic molecule where man can make whatever it wants to make. Right? And so therefore, N-acetyl glucosamine would have an acetyl group at the 2 position. Okay. Glycans can be modified with phosphate group, sulfate group, esters, acyl group, so many different types, and these are the ones that add to the structural diversity. And okay, I'm going to skip this part. Yeah.
So basically, with the AB, it just replaces the hydroxyl.
Yes.
And the name stays
The name stays same for any of. Yeah. Go ahead.
Is there, have there been studies where they modify the position of the amine and then see if that's also active or if that has different implications?
So obviously it depends on the system. The question is for Himangi and Melissa and team and Chelsea. The question is that have there been studies where the amine has been modified to observe a biological effect? And so it all depends on
position of amine.
Position of amine. Okay. Well, I don't really. Okay, so you move the amine from 2 to 3, and what happens?
Yeah.
Oh, that's a very interesting question. I don't think I have known much in that direction. So synthesis of carbohydrates, because that amine doesn't exist in nature, so you got to make it. Synthesis of carbohydrates is not everybody's cup of tea. It's an extremely challenging process, and generally, I would say that unless there is a good reason, people would not, try to investigate something of this sort. But it's something that you might want to focus on and develop in the future. Good, very good. Okay, so I'm going to skip this part because this slide is more for an sort of live lecture, and those people out there will not be able to follow as to what is going on here. So I'm going to skip this part, and it's a specific point about you as a student can actually find the stable, what is the stable conformation simply by drawing structures. Okay, you can actually do that. You can do this for salicylic acid. You can do it for so many other sugars. It's not very difficult. It is it's a logical progression of how the conformation could change, but it requires me to draw, and I think today we are not really capable of doing it, especially for the team in Wisconsin, New York, and DC. Okay, so I'm going to skip it for the time being. All right. Okay, let's do this. Let's pick up. So from that ends monosaccharide. Okay, that ends the section on monosaccharide. I'm going to begin disaccharide and polysaccharide next time. Okay, so we do have an hour on to do the structure. Any questions from you all? Okay, any questions from Wisconsin, New York, DC? Are you there?
Innova is good.
Okay.
No questions in Wisconsin.
All right. Thank you. I think we're going to end. Thanks for joining. Greatly appreciate it. Hope I was not speaking too fast and the words were not getting lost in, what is it, cloud.
I think it was great. I like the glucose. I was going to answer KEQ, equilibrium, pushes it the other way. So.
What equilibrium?
Your KEQ for your hemoglobin question. Your KEQ was pushing the open form. So.
It was on the other side, right? Was the equilibrium wrong direction?
Yeah. It was going to you have more going to the open form because you have more glucose because of the equilibrium thing. Okay.
The equilibrium is going towards the cyclic form. They have very little open chain form. Because there is very little open chain form, there is small amount of hemoglobin that is glycated. If you see significant increase in the small amount of hemoglobin glycated, that is most probably coming from a very high amount of the glucose that is present in the body, and therefore it is indicative of a diabetic or pro-diabetic state. That's the logic. Did you get that?
Yeah, I like it. I got that. Yeah.
All right. Very good. Excellent. I'll see you on Monday.
Yep.
Okay. Bye-bye.
Bye.
Okay. Thanks, guys.
Thank you.
Thank you.